Well, thank you, Jackie, for that kind introduction. And it's great to see so many people here to come out and support me with my inaugural lecture. There's quite a lot of people who said, it's very sort of, off-putting. And in fact, some people have tried to avoid it altogether. In fact, my old boss said, I managed to sidestep that one. I didn't do an inaugural lecture at all. So it's actually quite a privilege to actually see so many people here to come and talk to you about it today. Now, what I'm going to do, obviously Jackie said that I've been in research a long time now, so I'm not going to give you X number of years of research condensed into an hour. So I'm going to give you a flavour of some of the things that we've done. And a lot of people have said to me before about what is research actually like, and particularly research into Parkinson's, I say, it's like doing a 5,000-piece jigsaw puzzle, but nobody's given you the picture. So for years, what we've been doing is a slow, laborious bit. We're putting down the straight edges around the side. And as you know, that when you do a jigsaw puzzle, when you start to put down the centerpieces and the picture starts to emerge, then you can start to put the pieces down faster. And certainly, that's the stage that we are now. It's an exciting time for Parkinson's research. More is being done at a quicker rate. So what we'll do is really give you a flavour of some of the pieces of the jigsaw that we've helped contribute to the knowledge that's advancing Parkinson's research. All right, so I probably have to get my first slide up. Right, so you can also, just a few words about this. You can, whoops, I'm going backwards. Okay. You can also tweet, as well as the hashtag is Parkinson's. And you're probably thinking about why this strange title. And my illustrious colleagues who have gone before me know that the people who organise these things ask you, you've got to have a sexy, eye-catching title to draw people in. But there are actually more similarities than you think. So between a car crash and actually what causes and what things contribute to Parkinson's disease. So as I'll go through the lecture, I'll draw some similarities between the car crash. But first of all, before I start, I'd like to dedicate my talk to my partner. I know he's going to get embarrassed now, but he's been there supporting me through thick and thin. He puts up with me when my bleep goes off at 2:30 in the morning. And also to my mother, unfortunately, died, but I'm sure she'd be very proud if she was here. So let's look at the factors that contribute to a car crash. So here we have things like the driver is obviously a key component of a cause for a car crash. Obviously the age of the driver, obviously younger drivers are associated with more accidents. But who thinks women are better drivers than men? Ooh. Now, who thinks men are better drivers? Oh, obviously, yes. We're not going to go down that road, though. But certainly, the sex of the driver does play a role, and also the genetics as well. I think some people are naturally born to be drivers. A lot of people tell me that I'm a good driver, and I'm sure Fraser would agree that he was born to be a passenger. So he much prefers to be driven. So certainly there's a lot of things about the genetics. And also you've got the environmental conditions. Whether it's foggy, whether the roads are icy, whether there's huge amounts of traffic and it's very stressful driving, that can also account for things. And the biggest thing is probably the vehicle itself, mechanical failure. We have our MOTs to protect against this, but this is obviously a big factor in the car crash. So let's draw some of the comparisons with what we see in Parkinson's disease. Here you've got Michael J. Fox and Muhammad Ali, who developed Parkinson's disease, and you can see that there are multiple factors which are pretty similar to what you would see in a car crash. Certainly, as we go through, sex, age, stress, all play factors, and also the genetics play a factor. And one of the biggest things that we probably contributed to early on is our understanding about what goes wrong in the brain. So what is the mechanical failure? And once we have understood the mechanisms by which the cells are dying in the disease, then it's far easier to develop treatments for neuroprotection, which will protect against the neurons from actually dying. But before we start on some of the research that I've done, just to get everybody up to speed about what Parkinson's disease is. Now, it's a disorder, if you look about the general population, affects about 1 to 2 people in every 1,000 of the population. But as I said before, it's associated with age, and as people age, the percentage increases. So we get 2% of those who are aged 60 years and over, and this rises to 4% of those people who are over 80. So we've probably got about 130,000 people affected with Parkinson's in the UK at any one time. And depending on the sort of research that you read, this probably has an economic cost of about £3.5 billion per year. Now, the mean age of onset is about 65 years of age, but younger people can certainly develop the disease. And about 10% of those people who are diagnosed are actually under the age of 45, and the youngest person ever recorded was a girl of 13. Now, women are less likely to develop Parkinson's than men, and I'll come back to that topic later on. Now, what are the classical symptoms of Parkinson's disease? Now, most people recognize the tremor, and later on I'll actually have a video where a patient has a very marked hand tremor. So this is when the limbs are actually at rest. So when the person is sat in a chair with their hands in their lap, you might see the tremor in the hands. Obviously, the patients will actually complain that the limbs feel stiff and weak. And then you've also got what we call bradykinesia, so this is a slowness of movement. And also what you see in a Parkinson's patient, normally you've got a nice arm swing as people walk down the road, and this tends to disappear, and their arms are more trapped around the side, almost at a right angle. And later on in the disease, that they can sometimes develop postural abnormalities, so they're hunched over and they can have a short, shuffling gait. So that's some of the classical motor features of the disease, but unfortunately there are secondary, what we call non-motor features. So it spreads out of the motor pathways of the brain, and some of the common features that are seen in some patients are things like depression, pain in the limbs, taste disturbances, smell disturbances, and decline in cognitive function. Also, problems with sleep are also a common feature in the disease. Now, it also spreads out of the central nervous system into what we call the autonomic nervous system. So this does a lot of your body actions for you automatically, like your digestion, controlling your heart rate, and things like that. So this is called the autonomic nervous system. So the components of the autonomic nervous system which are affected, commonly you'll see constipation in most patients. You'll see problems with controlling their blood pressure, bladder problems, and impotence as well. Now, what actually is causing these problems? A lot of the pathological studies have now been completed, and we know what's actually going wrong in the disease. Now, one of the most common features of the disease is a degeneration in the motor pathways. And here you can see a line of darkly pigmented cells. And so these are cells which use the neurotransmitter dopamine within the brain. Now, here's a close-up, stained-up version, and you can see these peppered-like granules within the neuron. And this is neuromelanin. And so this gives the neurons are dark pigmented color, and when you cut through the level of the brainstem, you can see here the substantia nigra, even with the naked eye. And these neurons project to areas what we call the caudate and the putamen, and here the neurons release the neurotransmitter dopamine. So this is the communication system which really allows us to transfer our thoughts to actually picking up something to actually doing it. So here you can see the links. This is where you've got the basal ganglia pathways, and this is the nigrostriatal pathway here. So it allows you to not only initiate, control, but also to terminate movement as well. And here you can see a classical PET scan that we use to help diagnose Parkinson's patients. And this is really a radioactive tracer which will load up the dopaminergic nerve terminals within the brain. And here you can see that the caudate and the putamen are lighting up here nicely in the healthy subject with lots of sort of radioactive markers of the nerve terminals. And you can see in the Parkinson's patient, you've got much less signal here. Now, as I said before, you've got these non-motor features as well, because quite a few other pathways within the brain are affected. Both the noradrenergic and the cholinergic pathways are affected as well. So the pathology is also associated with a strange cytoplasmic inclusion called Lewy bodies. Now, these are these pink structures with this white halo around. And initially, it was thought that these structures were the bad guys in the equation that were possibly causing the neurons to die. But in fact, it may be that these are actually a protective mechanism within inside the cell. And these Lewy bodies are actually packed full of altered proteins. So these altered proteins are normally toxic to the cell, and one of the common ones that's found in Parkinson's disease is a protein called alpha-synuclein, and I'll come back to that a little later on. Now, there are drug treatments available for treating Parkinson's disease, but they all focus on the dopamine system. So all the therapies that have been generated so far target for replacing the lost dopamine. Now, you can't give dopamine itself because it can't cross this thing called the blood-brain barrier, which protects a membrane around the brain. Now, what they found was that the precursor to it, dopa, can actually cross the blood-brain barrier once you've given the tablet orally. So once it's inside the brain, it's converted via an enzyme, dopa decarboxylase, into dopamine. So the remaining dopaminergic neurons, you give L-dopa and it packs it full of the neurotransmitter dopamine. So once the neurotransmitter is released, it then sits on the receptors and it triggers the response in the adjacent neuron. Now, what I'll ask you to do is remember there's a little enzyme here called tyrosine hydroxylase, which in the normal situation converts tyrosine to L-dopa, and I'll come back to that enzyme in a little while. So other drugs which have been developed actually stimulate the receptors directly, so they don't rely on the integrity of the dopaminergic nerve terminals, and these are what's called dopamine agonists. And then the second sort of phase of treatments that were generated was that the neurotransmitter, once it's released and it's targeted its receptor here, then goes down a pathway of breakdown. And it's broken down by these cells called astrocytes and glial cells. And these have enzymes in them called monoamine oxidase and catechol-O-methyl transferase, which actually cause the breakdown of the neurotransmitter. So the next phase of developing treatments, we're actually developing inhibitors to stop the breakdown of dopamine, so more of it gets repackaged within inside the neuron. Now, the problem is with these treatments that they're effective at the very beginning, but they lose their effectiveness with time, and they're associated with marked side effects. And as I said before, that they only target the dopaminergic system, and as we've seen on the previous slide, many other pathways within the brain are also affected. So this is why when you give a patient a tablet of L-DOPA, you don't see a full functional recovery. And one of the big things is that these treatments don't stop the degeneration of the neurons. So really what we need to do is understand what is causing the neurons to die, and then we can develop therapies to block those pathways. Now, as Jackie mentioned before, one of the things that we've set up in the past is the Parkinson's UK Tissue Bank. Now, it's really important that we have human tissue to actually perform work on, because animals don't get Parkinson's disease. So there are strains of animals which are epileptic or they have problems with their blood pressure, so you can study them for those sort of diseases. But Mr. Rat doesn't get Parkinson's naturally, so looking at the tissue from people who've been affected by Parkinson's is again looking like the scene of an accident where you can gather vital clues as to what's actually going wrong. And when I was doing my PhD, we found it also quite difficult to obtain good quality tissue, and particularly without restrictions on its use. So this pipeline, as I'll show you later on, is actually vital. If we didn't have the human tissue, we're not really going to find out what causes Parkinson's. Now, in 2001, there was a call put out by the charity Parkinson's UK for people to actually establish a tissue bank. And fortunately, Richard Reynolds, who's over here, had set up the MS Tissue Bank already at Imperial College. So we had a very good platform, a very good established tissue bank in which to mirror establishing the Parkinson's one on. So together with Richard, we set up the tissue bank with a few other colleagues as well. And really, we set up with the aims of collecting high-quality tissue that had excellent clinical data on it, that it was neuropathologically characterized using the latest techniques. It was open access to everybody, and basically what we wanted to do is pump prime Parkinson's research. Now, we have what we call a prospective donor program. Basically, people carry a donor card like you would do a kidney donor card. And so these people have consented during life to donate their brain when they die. Now, when we first started off at the beginning of 2002, so we had no registered card-carrying people, and we had no brains coming into the bank. But Kirsten, where's Kirsten? Is she around? Kirsten Gormley? Yeah, right, she's over there. Who joined as a tissue bank manager with me then. We toured the country talking into patient groups, drumming up business, so to speak. And over the years, after Kirsten left, George has taken up the mantle as the tissue bank manager, and we still go and talk to patient groups. And now we have about 1,800 potential Parkinson's potential donors who carry the card with them. We have about 5,000 healthy people, because we also need healthy brains as well, because you need a comparison between what's happening in the disease to what's happening in a healthy brain. And over the years, you can see the red bars of the Parkinson's brains coming in. When we started off, we were collecting only a few brains, whereas now we're actually collecting large numbers of brains every year. So we supply tissue all over the world to different projects, and so this is vital for research. Okay, so enough of the sales pitch about the tissue bank. So let's talk a little bit more about some of the research. So let's take some of the similarities about sex, about stress, and the genetics. Now, your genes, and these are not your 501 style, so these are your DNA genes, and these are really vital for cell survival. Now, you can have transcription factors, which then come in and interact with the DNA, and then you get the replication and you get the formation of proteins. Now, these proteins are essential for the survival of your cells. Now, it was first thought that genetics really didn't play a major role in Parkinson's disease. But there were several groups of people dotted around the world where there were small families of where it appeared to run in the families. And with the explosion in really the abilities to gene sequence after the whole genome was sequenced, it was been found by other groups around the world that there are what we call gene mutations and defects in the genes which lead to defective proteins being formed. And now there's probably about 16 gene mutations that have been detected in Parkinson's cases. And these are what we call familial Parkinson's, but they only count for a very small percentage, and some of these gene mutations are very rare indeed. So what we've done is really try to concentrate, what about the sporadic cases? So these are the ones that are not associated with a gene mutation. And what we've done is teamed up with other researchers around the world looking at genome-wide association. John Hardy at UCL set up an international Parkinson consortium. So where we donated all our samples from our cases coming into the tissue bank, and we did these genome-wide association studies. So all in all, samples across the world, and we ran these chips on about 8,500 Parkinson's patients. And it came out with some interesting, what we call SNPs, or slight alterations in the gene, and these are called polymorphisms. And there was 15 risk genes which are identified. Now, this research is still in its infancy. We don't know exactly how these risk genes actually operate and what degree of risk that confers on you. But certainly it doesn't mean to say that if you've got these risk genes that you will actually develop Parkinson's disease. You must be exposed to several other factors before you develop the disease. Now, there's also one other way that you can alter protein formation, and this is an area what we call epigenetics. Now, this is quite a new and exciting area, and the epigenetics means that it is above the genome. And this can cause quite marked changes in gene expression, which can also have marked changes on cell survival. Now, these are some nice pictures taken from a review that Ian Harrison in my group has actually written. Now, your DNA inside every cell of your body, if you just let it loose, it will be very long and it would get in the way of everywhere. So what we find is that it's coiled tightly around these proteins which we call histones. Now, there are enzymes within the cell, these HDACs, which are these histone deacetylase enzymes, or these histone acetyltransferase enzymes. And these really regulate how tightly wound your genes are. And this really does dictate how your transcription factors can actually get in. So if your DNA, like this ball of wool, is very tightly bound, and this is my transcription factor, you can see that it can't get in there to trigger transcription of the genes. Whereas if you've got loosely bound DNA, you can see that the transcription factor will get into the genes and cause new proteins to be synthesized. So what we think is that normally there's this nice balance between the HDACs and the HATs, so it allows cells to survive. But we think that in neurodegenerative diseases like Parkinson's and many other diseases, that the HDACs become overrepresented compared to the HATs. And so what we've done recently is look at enzymes which could block these HDACs. And so basically, rather than tightly coiling the DNA, it can leave it more in a loose structure. Now, Ian has been working on this, and we've looked at several compounds already, and I'm going to show you just some data from one of them. This is Valproate, and this was developed in the 1970s as an anti-epileptic treatment, and is used to treat partial and full seizures in not only adults but also in children. And later on, it was found that Valproate, if you put cells into a culture dish and exposed them to Valproate, it actually had a positive effect on their survival. Now, these HDAC inhibitors, like Valproate, could not only have a beneficial effect on the neurons, preserving their survival, but it also can have beneficial effects on the other cells within the brain, such as the astrocytes and the microglia, dampening down inflammation or promoting the secretion of growth factors, which are necessary for the neurons to survive. And it's been proposed that compounds like Valproate have an additional activity by inhibiting these HDAC enzymes, and so that they leave the DNA in a looser coil, allowing the transcription factors to gain better access. Now, as you can see here, we've got MRI images, and this has been used quite extensively in medicine for the diagnosis and also for the monitoring of different diseases, but we can also use MRI within animal models. And so Ian, this is one of his scans of a rat brain in the MRI scanner, and what Ian has done is done a study looking at Valproate. And what we've done is used an animal model where under deep anesthesia, a toxin is injected onto one side of the brain. Now, as I said before, animals don't get Parkinson's disease normally, so you have to inject toxin into the motor area of the brain in order to make it Parkinson's. So you're just doing this on one side of the brain. So if you pop an animal into a clear plastic cylinder, what it will do is use its paws, it will rear up to try and look to get out. Now, a normal animal will use both its left and its right paws as it's exploring the environment. But as the toxin starts to work and the animal becomes a Parkinson's model, what it will do is just use its its left paw and leave its right paw fairly close to its chest. So you'll see a difference in the paw usage. And also, what you can do, amphetamine is a sort of an illegal drug, but if you give people amphetamines, they become hyperactive. And if you give a rat amphetamine, it'll become hyperactive. But because you've damaged the motor pathways on one side of the brain, the rat will actually walk around in a circle rather than just being hyperactive, walking normally in a cage. So you can use these behaviors to actually measure whether the drug is actually having a neuroprotective effect or whether it's not. So we have a baseline, we do all these MRI scans and behavioral tests, and then we administer the toxin. And then what we've done is a week later, we've started the Valproate treatment. So the cells are well on their way to actually dying. And so then we administer the Valproate and then measure for a few more weeks to see whether it's protective. Now, what we'll do is take this slide fairly slowly. You can see the top lines here, the red and the blue. So this is animals that haven't had any of the toxin, the lactocystin, injected into their brain. You can see that they're using 50% almost right and left forepaws as they're exploring their environment. Now, the green line is the animals that have had the toxin, that are becoming a Parkinson's model, and as you can see over time, that their right paw stops being used and they're mainly using their left paw. Now, after 1 week, where we start the Valproate treatment, this is where it becomes interesting and where we can see the purple lines and the yellow lines here. Hopefully you can see them better than I can, but the purple line is where it's a low dose of Valproate and the yellow line is a high dose. And as you can see here, that the purple one has a beneficial effect. Here's your lactocystin animals alone, low dose of Valproate, and an even higher one, it actually returns the animal back to normal behavior. And here you can see it in a graphical form where we've looked at the area under the curve here. And as you can see in the green bars, you get a progressive increase in deficit in the Parkinson's animals. And then you can see this being restored with the Valproate treatment. And this is also true for the number of dopaminergic neurons remaining in the brain. Here you can see this is the percentage loss of tyrosine hydroxylase positive cells. And this is, I said, to remember this enzyme, and you saw it in the synthetic pathway that makes dopamine. So what you can do is use a colored tag to mark this enzyme with inside the neurons. So you can visualize the neurons when you look at them under the microscope, and it allows you to count them. So this is the percentage loss of dopaminergic neurons. You can see there's no loss obviously in normal animals, but in the lactocystin lesioned animals, you can see there's quite a marked loss of these cells. And in the animals that have then received the Valproate treatment, you can see a steady protection in these animals. So that's all very well and good. And what Ian is doing at the moment is actually trying to look at the MRI data by segmenting out the different brain areas and calculating the volume. Also, we're doing some work with Bill and Anthony at King's, who have got a lot more whizzo about the computing power, and they can do this using what's called voxel-based analysis. And you can see here that you get patterns of changes within the lactocystin-treated animals. And so what we're doing now is actually looking to see where the Valproate improves that. So this is an ongoing project. So the beauty of these things is that if Valproate is actually protective, it's been used to treat epilepsy patients for many years, so it could quickly progress into being tested in Parkinson's patients. Now, this is some work that we did with Glenda Gillies, and really it's looking at the sex differences within Parkinson's disease. Now, epidemiology studies clearly show that there is a difference in the incidence of Parkinson's disease in males and females. And here you can see the men and the females at different ages, and there are clearly differences in the incidence. Now, also, if you have a very young Parkinson's lady who is still going through the estrous cycle, she actually responds differently to her drugs at different stages during the cycle. And so when the hormone levels are fluctuating, it indicates that it is actually affecting the response to the drugs. And also, if you have a slightly older lady who's gone past the menopause but is also on hormone and replacement therapy, if you take away the hormone replacement therapy, the Parkinson's actually gets worse. So this gave us a strong indication that the hormones may have certainly a significant role to play. Now, the other model that we've used quite regularly is the 6-hydroxydopamine model, and this is again a toxin that you have to inject when the animal is deeply anesthetized. So you make a small injection deep into the brain to target the motor pathways. And here you can see the tyrosine hydroxylase positively labeled cells on the healthy side of the brain on this side, whereas the ones where you've injected the toxin, you can see that there is a marked loss of dopamine cells within this model. So what we've done here is actually compare this model in males and females. And so we've used different doses of the toxin and looked at the percentage of the cell loss. And here you can see the percentage survival of cells, and as you can see that as you increase the dose, you get less survival. But what you can see straight away is that there is a big difference between the percentage survival in the male animals compared to the females. Certainly there is a greater survival of cells within the female brain compared to the males. So this indicates to us that some of the female sex steroid hormones are having a protective effect. And just to sort of emphasize this, if you take female animals at different stages of their estrous cycle, now the estrous cycle in the rat is actually quite short. It's only 4 days. But if you take the animals that when they're at proestrus, i.e. there's an estrogen surge, and you compare them to animals when it's at diestrus, i.e. when the estrogen levels are low, and you expose them to the toxin, you can see that there is a difference again. So when estrogen levels are high, you get protection, and when it's low, you start to see more cell loss. And then we also did some quite clever experiments again with Glenda Gillies. And this time, we actually compared males and females, but what we did was we took out the ovaries or the testes. So the ovaries actually produce estrogen within the body. So here you can see the intact males and females, and this is the percentage loss of dopamine this time, which is the neurotransmitter within the brain. And you can see that when you expose these animals, you still see this male-female difference. So let's first just concentrate on the females. If you first of all take away their ovaries, allow them to recover from the operation, and then later on you expose them to the toxin, you can see that the loss of dopamine within the brain is a lot less, so indicating that estrogen is actually protective. Now, if you put back the male sex steroid hormone testosterone, you don't see any protection. But in another group of animals, which we took the ovaries out, this GDX group here, and then we put back slow-release pellets of estrogen, you can see that you can restore this level of protection. So again, indicating that it's estrogen in these animals which is actually protective. And so we thought, well, can you see the same in the males? And if you take away the testes, which produces the testosterone, you can see that the survival of the dopamine levels within the brain is actually much better. So it indicates that testosterone in the males is actually having a negative effect, whereas estrogen in the females is having a protective effect. However, we saw something quite strange, that when we gave the male animals estrogen, it didn't have a protective effect in them. So it indicates to us that when the brain is being masculinized or feminized, the response to estrogen is programmed in when you're very young. And in fact, it's probably that the male brain is responding differently to the estrogen. Certainly, it's very interesting to see that estrogen is actually protective in the females. So that's an interesting step. And also what we've looked at are some of the environmental factors. And when they've done these large epidemiology studies, they found that stress has been quite significantly associated with Parkinson's disease in many epidemiology studies. Now, stress is normally a protective response, and it involves this, what we call the HPA pathway, the hypothalamus, the pituitary, and the adrenal cortex pathway. And you end up releasing corticosteroids, which normally allows you to survive the stressful period and then overcome it. But the problem is that in chronic stress, this high level of corticosteroids can actually be damaging. And certainly, if you restrain stress animals, you can get the increase in glucocorticoids can actually result in a damage of an area that is associated with memory. And also, it also induces a process called oxidative stress, which I'll come back to in a short while. So what we wanted to do is to see whether, if you stress, whether it actually makes you susceptible to Parkinson's. And we did this by looking at a study. And there's one other thing as well, that the corticosteroids are actually used to treat mothers in pregnancy to try and improve the survival rate. So one of the worrying things was, if you give corticosteroids to people, does it leave them vulnerable to Parkinson's disease later on? So we split up this complicated study into two sections. So we gave some of the animals while the mother was pregnant dexamethasone in their drinking water, and some of them had just nothing in their drinking water at all. And they allowed them to grow up to adulthood. And then some of them were exposed to what we call restraint stress. Now, this may sound a bit awful, but all the animal does is actually go into a clear plastic tube. The animal knows that it can't get out. It stays in there for a while, and obviously this is a mild stress for the animal. and afterwards we just let the animal out. There's no harm to the animal. And then later on in their life, what we do is expose them again to a small dose of the toxin to see whether this has stimulated any changes. So again, what I'll do is walk you through the slide. So this time, you're comparing the lesion side of the brain with the unlesioned side of the brain. So these dark orangey yellow bars are the unlesioned side, whereas the yellow ones are the lesion side. And this time we chose a dose of the toxin that only produces a very small amount of cell loss. And here you can see the control animals which have just received the 6-hydroxydopamine. You can see a small cell loss here. Now, if you expose the mother to the corticosteroids during pregnancy, this doesn't have any additional effect on it, which is very good. But with the male animals, when you expose them to stress over a period, and then you allow them to get older, and then you expose them to the toxin, you can see that the cell loss jumps from here down to here. So the stress does predispose them to the toxic effects of the 6-hydroxydopamine. Now, the story in the females was very different. So again, the toxin just causes a very small reduction in cells. But when we restrained stress the females, it was actually protective. So again, this highlights that there is this sexual dimorphism in the vulnerability of the motor system to toxins. And it may be that in the female brain, that because they're exposed to high concentrations of estrogen every month, and this will act as a stressor to the brain, that the females are used to handling stress, and it can actually be a protective mechanism. And it goes through the old adage that females are far better at coping with stress than males. And what we want to do now is take this further forward and investigate why in the males they're very susceptible to the effects of stress. Why is it making them more vulnerable to the toxin? So again, this is an ongoing project, but it's a very important finding. As I said before, one of the areas where we have made quite a lot of contribution is actually understanding what goes wrong in the brain to cause Parkinson's disease. And then once we've done that, we've developed drugs to try and stop the disease. Now, this is where this pipeline approach to work on human tissue, finding out something that's gone wrong in the tissue, testing it in our animal models, and then going into the clinic has actually been very fruitful. And I'll show you one of the incidences where we've moved from here right into the clinics. First of all, I'll take you right back to when I was doing my PhD with Peter Jenner's group at King's. And one of the things that we looked at was iron in the brain. Now, you need iron inside your cells to function. And what we did was we looked at the substantia nigra from healthy brain donors, and we looked at the substantia nigra that was left in the Parkinson's brain. And what we found is that in the Parkinson's brain, they had a lot more iron in them. And so what you can see here, that it's bad for the cells. But the thing is, why is it bad? This element that is around us all the time, we use it in our everyday living. It's not only allows us to get around on the tube, make our toast in the morning, but it's actually vital for all our cellular functions as well. Now, our mitochondria are these strange structures here, and these are our powerhouses inside the cell. So they make all the energy that is required for the cell to survive. And we all know about our red blood cells, which are packed full of iron. They carry our oxygen around. The iron is also very vital for helping synthesize neurotransmitters and actually the neurotransmission between neurons as well. So why is this essential element potentially damaging? Now, iron is a bit like a Jekyll and Hyde character. It has a good side, but also it has a very bad side to it as well. Now, within the mitochondria, we use oxygen to produce energy, and our energy packets are called ATP. And as a byproduct from this energy production, we get a molecule, what's formed, which is called superoxide. And this in itself isn't too harmful. We have mechanisms, we have enzymes like superoxide dismutase, which convert it to hydrogen peroxide, and then catalase then converts it harmlessly to water. Now, the problem is that if we've got a lot of iron floating around, it can interact with the hydrogen peroxide to produce what we call the hydroxyl radical. Now, the best way of telling you what a radical is, it's basically like a politician. You know, they can't leave anything alone. And here's your naughty radical on the top of the slide. And normally you have electrons spinning around the outside of the molecule, and normally they go around in pairs. And when they're pairs, they're nicely balanced. But when they're unpaired, like this guy here, he's only got 5, so he's got an outer electron which is unpaired, so he's unstable. So what does he do? He takes an electron from an adjacent molecule to gain stability. But in doing so, he damages this molecule here. So if it does it around your lipids, your membranes, things like that, your DNA, this can be very bad news for the cell. So this is why iron can be toxic. So in fact, what we showed that not only iron is potentially generating oxidative stress, but also we looked in the Parkinson's brain and showed that there was a lot of free radical damage to things like the lipids, the DNA, and the proteins. So this can have a direct effect on cell survival. But there's one of the other things that it can also do is it can have an effect on proteins. Now, here we're talking about this protein that is associated with Parkinson's disease called alpha-synuclein. Normally in the cell, it's unfolded. But what can happen is, if you've got free radicals and oxidative stress, it can cause the protein to aggregate, to misfold, and then it can cause toxicity inside the cell. Now, normally you have a mechanism called the proteasome, which normally gets rid of altered proteins. But also what other groups have shown is that the proteasome is malfunctioning within the Parkinson's brain. So not only have you got an increased capability of producing these altered proteins which cause toxicity, but your mechanism, your mincing machine that gets rid of these altered proteins is actually dysfunctional. So you get more altered proteins being formed. So this is now a fairly commonly recognized sort of pathway of toxicity inside the cell. But can we do anything about it? The iron is here, but can we get rid of it? So what we've tried to do is actually to see whether drugs called iron chelators are actually potentially useful for Parkinson's disease. Now, in order to study this, you need a model of where the iron overload is in the brain. You treat the animal with a chelator and see whether it pulls the iron back out again. Now, you can't simply put an animal on a high iron diet because it doesn't load the brain. You've got the blood-brain barrier which stops it from loading the brain. Now, my colleagues that I worked with in Belgium, Bob and Bobby, we developed this ferrozine-loaded animal model of brain iron overload. This ferrozine molecule has an iron molecule embedded in its structure. And it's very lipophilic, so it crosses the membrane, gets into the brain, it gets metabolized, and then it drops the iron into the cells inside the brain. So you can put the ferrozine into the diet. And so if you put the animal on a normal diet, this is your level of iron. If you put it on a ferrozine-loaded diet, the iron levels go up quite considerably. And so there are iron chelators which are around and are used for treating other diseases. And together with Bob Hyder and other people at King's, we tested some novel chelators as well. And just over 2 weeks, if we gave 6 doses of the chelator over 2 weeks, you can return the brain iron content back to what it would be in the control animal. So again, this gave us a good idea that these chelators might be useful. So then we tested them in our Parkinson's models. Now, there are 3 commercially available iron chelators that are used to treat a disease called beta thalassemia. So these patients have defective red blood cells, and so they need repeated blood transfusions in order for them to have a healthy lifestyle. The bad thing is that when the body breaks down the red blood cells, liberating the iron, there is no mechanism of getting it outside the body. So these beta thalassemia patients, the iron accumulates in the body, particularly in organs like the heart, and causes heart attacks, and these patients don't have a long survival if you don't actually treat them. So these iron chelators have been around on the market for a number of years, particularly desferrioxamine has been around for a number of years, and they're very successful at treating these patients because they grab hold of the iron, they envelop it, and then they take it out of the body. Either you pee it out when you go to the toilet, or it goes out through the digestive system. So what we thought was, can we test these chelators in our Parkinson's models as well? So again, this is the 6-hydroxydopamine lesion animal model, where you've got the toxin just on one side of the brain, the other side of the brain is actually entirely normal. And so here you can see the loss of your tyrosine hydroxylase or your dopaminergic neurons within the model. So this is the animal that's just had the toxin, and you can see that you're killing off probably about 45% of the cells here. So we've given the toxin, we allow them to recover for 2 days, and then we start the chelator therapy. And here you can see that just with 7 days of chelator therapy, the loss of cells is actually considerably less. In fact, with this drug here called deferroprone, you can see that the loss of cells is actually much less than what you would see in the vehicle-treated animals. Now, when we go to look at the neurotransmitter dopamine within the brain, you can see here in the animals that have just had the toxin, there is a marked loss of dopamine within the brain. And so when you give the chelator, particularly deferroprone, you can see it almost entirely protects the dopamine within the striatal system. So what we've done now is actually move this into the clinic. And so what we're running at the moment is a double-blind, placebo-controlled study looking at Parkinson's patients. So we've got a randomized, placebo-controlled, double-blinded study, so we don't know what the patients are taking, and they don't know what they're taking. So we've got lots of them taking the compound, and we assess their Parkinson's function, and we're also looking at their memory, their depression rating, and also their quality of life, and we're monitoring their brain iron concentrations. So this is again ongoing. So what we hope to do by the end of the year is actually report about whether this drug is actually worth taking forward into a larger study. Okay, so it's not only drugs, but also surgical approaches have become very prominent in potentially treating Parkinson's disease. And this is Mike, who's had deep brain stimulation put in here. You can see the power pack in his chest going up and having the electrodes going down deep into his brain. What I'll do is I'll just play this clip, a very short clip, and then...
The probe that is placed into the brain, I understand...
So this is with his stimulator
with the
on.
top of the nose and the ear. And it is connected to a battery in my chest, on the skin in my chest here. And this is controlled. I can control the amount of the voltage going in, I can control the length of time that the pulse is, and I can identify the number of times per second as it goes in. And I'll turn myself off now.
So as you can see, he's got a very marked tremor here. And so the deep brain stimulator is controlling his tremor. So the thing is that why this is actually occurring is that the loss of dopamine within the nigrostriatal system, it regulates a lot of other pathways within the brain. And one of the pathways becomes overactive. And this pathway uses the neurotransmitter glutamate. So glutamate is an excitatory neurotransmitter. And it's this that really ultimately causes the Parkinson symptoms, because it actually in the end blocks the translation of your thoughts to doing something to actually translate into pick up an object or actually move. And the thing is, this is actually done under stereotactic surgery when the patient is awake, but not every patient is actually suitable for DBS therapy. So what we've tried to do is to see whether we can actually mirror these effects of deep brain stimulation using drugs. Now, unfortunately, it's a complicated system, but here is your glutamate nerve ending. It releases glutamate, and it triggers these receptors on the target site. Now, the main types of receptors are these inotropic ones, and basically it does what it says on the tin. If you trigger these receptors, it lets ions inside the cell, and then it causes a depolarization within inside that cell. Now, certainly, drugs which block these receptors, so they block the responses of glutamate, are certainly neuroprotective. But when it came to actually work in humans, it found that these drugs are associated with lots of side effects. So what we've done is actually looked at another group of receptors which can control the glutamate levels, and these are the metabotropic ones. And there are 8 metabotropic glutamate receptors. Some of them are located presynaptically, and they'll slow down the release of glutamate, while other ones will actually enhance the effects of glutamate on the target cell. So really, just to cut it short, because time is clear, is currently a PhD student, and Anthony Vernon started this work off in my lab many years ago, and Claire has taken up the baton in this work, basically looking at our Parkinson's models. Again, lesioning the animals, allowing them to recover for a while, and then starting the drug treatments. You can see here, this is the cylinder test where you've got the pause being used by the animals as a gradual decline in the use of the pause within the cylinder test. And if you treat with the drugs, that you can restore this behavior. And certainly within the treated groups, in this one it's DCPG, which is an mGlu-8 agonist. Again, you can see protective effects. Now again, in another drug, now all the old style types of drugs went directly for the receptor itself. And this caused problems because it was very difficult to design these drugs that would get across the blood-brain barrier. So this whole field was very slow for a number of years until they designed what's called allosteric modulators. And these fit inside the actual molecule itself within the receptor and don't bind to the receptor site itself. And so they cause a more subtle modification of the response of the receptor. But these drugs are much more lipophilic, and they get across into the brain more easily. And this is some work that was done by Hugh Chan in my group, which is an mGlu-4 allosteric modulator called PHCCC. And again, you can see here, if you look at the cell loss within the brain, you can see within the lesioned animals you get a marked cell loss, but if you treat them with PHCCC, you get a protection. So we're still searching for which is the most ideal form of this drug to actually take into a clinical trial. So what I'll do is, time's pushing on, so I'll skip this group of slides and move on to, as Jackie mentioned, not all my work has been hard science. What we've had is some fun down the line. I think Elaine is around. Where's Elaine? We had some number of years working at the Dana Center together, running public forums for debate, particularly in neuroscience. And so we ran interesting events like looking at how taste evokes things in the brain. We've also worked with a theatre company called Y Touring, and they produce plays which go around going for schools. One of the ones which I worked on in the past was one called Mind the Gap, and they've got a new one which has come out, which actually involves deep brain stimulation. This one is stunted trees and broken pathways. I've also worked with artists, particularly with Robert at GV Art. And here we've got artists who came along and saw brain dissections. And then there was artwork which has been generated from that. And this was displayed at an art exhibition called Brainstorm. We also contributed things for the Brains exhibition, which was run at the Wellcome Trust in 2012. And recently, we've been doing some work with Susan Aldworth, who currently has a very nice exhibition at the National Portrait Gallery, where she produces amazing prints about epilepsy patients. And what we've been doing with Susan, she's very interested in printing. And we've done some pilot studies where we've actually contacted brain slices onto lithographic print plates, and then we can actually print from these plates. And here you can see some very interesting images. And from these pilots, we then applied for an Arts Council award to take it further forward, and which has now been granted, so Susan can do the full brain slices that we've got and get some of these amazing slices. So she's got an exhibition of these slices once it's all finished at GV Art, which is going to be on the 5th of June. And then also they're going to be displayed here at the Blythe Gallery, Imperial College, later on this year. This just leaves me really to just thank all the people who've done all the hard work. I did quite a lot of work, lab work, in my earlier years, but now you move out of that, and then you have people coming in, some exciting scientists coming in and working from you. So these are people, past and present, who have worked with me, my colleagues at Imperial College, who I have healthy interactions with, and also the people outside the college. And we've been very fortunate attracting different amounts of funding from a wide variety of organizations. So I'll close there.